Okay, good morning, students. I hope I'm audible.
Good
Yes,
morning.
ma'am. Audible.
Good
Okay,
morning.
so in the first group, we have enrollment number 2, 3, 4, and 5, right? So can we have the presentation from the first group? Yes,
Excuse
what?
me, ma'am.
Yes,
Uh, this Taiba this side. Ma'am, I was hoping if my group can go second. We are,
your group is otherwise?
it's group 4.
Yes, you can do it. Let's see if second group is ready. There's one group which is not ready, the Athoiba group. So instead of that group, you can do it, right?
Ma'am, excuse
Okay,
me.
ma'am. Thank you.
Yes.
Ma'am, excuse me.
Yes, Devanshu.
Ma'am, I'm having exam after this class. So can we go second? We have the serial number 3.
See, you cannot, but you have to be in the. See, I told you the marks will be given on the basis of the questions asked. So even if you have you presentation is done, you are required to stay back for others' presentations so that you can ask questions, right? I've mentioned this in the assignment. So even then you need to be that. Which exam you have, Devanshi?
Ma'am, the medical exam.
Medical exam?
Ma'am, I filled an exam for police, so that's why I'm
Okay, fine. That can be taken, right? Fine, fine, Devanshi.
okay, ma'am.
Yes, please switch on your videos also. Sam is going to present first, right?
No, ma'am. Tridev is going to start with introduction.
Okay, Tridev, fine, good.
Good morning, ma'am.
Good
My
morning. Good.
switching. Good morning, ma'am. So the topic of our presentation is introduction to plant, human, and animal viruses. So I will be presenting the slides containing origin of viruses and history and whether the viruses are living or non-living organisms, how they form the connection between living and non-living organisms. So viruses are basically a simple and acellular infectious agents. Viruses are microscopic, obligate cellular parasites which generally are much smaller than bacteria. They lack the capacity to thrive and reproduce outside the whole host body. So, what is actually the origin of viruses? The origin of viruses is, however, unclear because they do not form fossils, and that's why there are three main hypotheses that aim to explain the origin of viruses. First is the regressive hypothesis, which is also known as the degeneracy hypothesis and reduction hypothesis. In this hypothesis, it is claimed that viruses are small cellular organisms that parasitize larger cells. Over time, however, genes not required by them and they become simpler organisms, which are the viruses we see today. Next hypothesis is the escape gene hypothesis, and in this, some viruses may have evolved. This claims that some viruses have been evolved from bits and pieces of DNA that escaped from the genes of larger organisms. The escaped DNA could have come from plasmids and ontrosporons. This is sometimes called the vagrancy hypothesis or the escape hypothesis. Next hypothesis is the co-evolution hypothesis. This is also known as the virus first hypothesis. This hypothesis proposes that viruses may have evolved from complex molecules of proteins and nucleic acids at the same time as cells first appeared on Earth and could have been dependent on cellular life for billions of years. Sam, next slide. Next is the history of the viruses. Uh, there is a branch in science which deals with the study of viruses, and then that branch is known as virology. The term virus has been derived from a Latin word referring poison and other noxious liquids. Viruses can infect all type of life forms, including multicellular organisms and unicellular organisms. Now, coming to the history. In 1984, there was a French microbiologist, Chamberlain, invented a filter with pores. However, the pores were very small and even smaller than the bacteria. Thus, he could pass a solution containing bacteria through the filter and completely remove them. In 1992, there was a Russian biologist, Dmitry Ivansky, first discovered the virus in an infected tobacco plant. In 1998, the Dutch microbiologist Martinus Beijerinck observed that these agents multiply uh, only in cells that are not dividing. He called it a contagium vivum fluidum and reintroduced the word virus. The tobacco mosaic virus was the first to be crystallized by Wendell in 1935, and its structure could therefore be explained in detail. By coming to the end uh, of the 19th century, however, Sam, next slide, please. By coming to the end of 19th century, viruses were defined in terms of infectivity, ability to be filtered, and their requirement for living hosts. Viruses had been grown only in plants and animals. The early 20th century, in the early 20th century, the English bacteriologist discovered a group of virus that infects bacteria, now called bacteriophages. In uh, the second half of 20th century was the golden age of virus. Discovered the most of the in the discovery of most of over the year of 2000 recognized species of animals, plants, and bacterial viruses were discovered during these years. Sam, next slide, please. Characteristics and properties of viruses. Viruses are cellular and non-cytoplasmic infectious agents. Therefore, a unit of virus is referred to as virus particle rather than a virus cell. They are smaller than bacteria and can pass through bacteriological filter. They are consisting mainly of nucleic acids surrounded by a protein envelope called capsid. They are devoid of the sophisticated enzymatic and biosynthetic machinery essential for the independent activities of cellular life. Therefore, they can grow only inside the suitable living cells. These viruses do not grow, neither respire nor metabolize, but they only reproduce. Viruses may even be crystallized much like molecules, although some kind of viruses can only be purified but not crystallized. Sam, next slide. A virus cannot contain both DNA and RNA. Therefore, a virus can only be a DNA virus or an RNA virus, depending whether they contain nucleic acid DNA or as RNA. Viruses are transmissible from diseases to healthy organisms. All viruses are obligate parasites and can multiply only within the living host cells. Viruses are host specific, that they infect only a single species and definite cells of the host. They are highly resistant to germicides and extremes of physical conditions. Viruses are called the connective link between living and non-living organisms. Now, uh, uh, Sam, next slide. As it is said, it is a connective link between living and non-living organisms. Let's see why do we actually consider viruses as a connective link between living and non-living organisms. Viruses are termed as living organisms because they possess a genetic material. However, it's also considered a non-living because they do not crystallize. Then they undergo mutation. That is another characteristic of a living organism, but they are inert outside the host body. That's why again a non-living characteristic. They show irritability, and this is again a living thing, but lacks cell membrane and cell wall. That is not a living thing. That is a non-living characteristic. They are capable of reproducing, and they increase in number. So they are considered living, but lack of cytoplasm and organelles, they do not show cell division. That's why again considered as non-living. They can be transmitted from one host to another. They react to heat, chemicals, and radiations. They develop resistance to antibiotics. All these characteristics are fulfilling the criteria of viruses are living, but however they do not grow in size or shape, they do not possess sort of nutrients, they do not respire and excrete, they undergo their own metabolism, lack any energy producing system. All these characteristics refer to them as non-living organisms. So it builds a connection between living and non-living organisms. However, they cannot be particularly classified. Sam, next slide. Now my friends will elaborate.
Okay, thank you, Sudev. I'm just switching on my camera. One second.
Just a bit, Manaswini. I want to ask Tridev because I will keep asking question just after one's turn is over, right? So Tridev, my question is that you have said one particular thing when you were saying differences between viruses, like living or non-living, that they develop resistance to antibiotics because viruses cannot be cured by antibiotics. So I feel there should be antivirals there, not antibiotics. Otherwise, because viruses, antibiotics don't work on viruses, right? So please make sure you clarify this term, right? Are you getting my point?
Yes, ma'am. Yes.
है ना? My next question is, what are Chamberlain filters? You mentioned about the Chamberlain filters in the beginning. Can you just tell me what are those filters?
Ma'am, those filters were like, it was made by the person Charles Chamberlain. That's why
Mhm.
it's named as Chamberlain filters. And ma'am, he made a filter in which he passed a solution of bacteria, and the pores were small. So
Mhm.
the
Yes,
bacteria to
were
basically
retained.
remove the bacteria, ma'am.
Yes,
Like
yes, yes. These were the porcelain filters made of a very small pores and which could retain the bacteria, right? Okay, Tridip, can we have a next student now? Good, Tridip, it was a good present.
Okay, so good morning. So I'm going to be talking about a little bit about plant viruses, but that won't be the only thing that I'll be focusing on. I'll also be talking about, we'll see. So anyway, I'm starting with my part. So first of all, let's talk a little bit about the structure of plant viruses. So like Tridiv had mentioned, I'm going to just give you a basic definition of the structure of a virus. So any virus in the simplest terms can be defined as a genetic material that is surrounded by a protein coat or a protein shell. So they are very, very small organisms, and which is why even compared to a bacteria, they are very, very small. So the typical diameter of, let's say, any spherical plant virus is around 30 nanometers. So an example of a spherical plant virus can be the apple latent spherical virus, which obviously, as it's mentioned in the name, it does affect the apple plant. Now, let's talk a little bit about the general structure. So, like I said, they have a genome and a protein coat. So, the genome can be either RNA or DNA, and the protein coat is something called a capsid. So, a capsid is held together by these small other polypeptides called capsomeres, and they are the ones which, in a way, configure the symmetry of the virus, which we will be looking at in the next slide. So, we have a capsid, which is a protein coat, and then we have either RNA or DNA. So this entire unit, let's say it's infectious, which it most likely is, then it's called a virion. So a virion basically, again, in some cases, a virion can have a few extra components like a glycoprotein spike or an envelope, and in some cases even enzymes. So retroviruses have the enzyme reverse transcriptase, as we all know. So like that, it does vary in some viruses, but all of them have this same general structure. So like I had mentioned, there are capsomeres which make up the capsid, and these are oriented in different symmetries. So some of these symmetry, the most common symmetries are helical and spherical. There are also filamentous or rigid rod-shaped viruses, and there is also icosahedral symmetry. So an icosahedron is basically a figure with 20 plane faces, and each of those faces is an equilateral triangle. And there are also some viruses which have something called a complex symmetry, which doesn't exactly fit into any of these aforementioned symmetries here. So an example of this is a Gemini virus, and as we know, Gemini virus is a plant virus that infects many, many different kinds of plants and causes some very severe infections. So Sam, next slide. So like I've just mentioned here, just a few examples of some of the common viruses. So we've got the rod shaped one. As you can see, it's very rigid. And then we've got spherical shaped. And the Gemini virus, as you can see, it looks slightly spherical, but you can't really club it against spherical shape because, as you can see, it's still a bit of a distorted shape. So Sam, next slide. So let's talk a little bit about the properties of virus particles. This does not apply to just plant viruses. I'm talking about viruses in general. So first of all, like Trituv had mentioned, the infectiousness or the infectivity of the virus depends on the host system. Why? Because the host is the one that is actually producing the viral proteins and the virus particles, right? So once the virus actually invades the host cell is what is responsible for in a way furthering the infection, because they are the ones who become like a kind of virus factory. So this is why you can say that viruses are in a way cellular obligate parasites. Obligate because they can survive only within the host cell, they cannot survive on their own. So all of them have a few general properties. So one of these is that they all thrive in a certain optimal temperature. All of them don't thrive under the same temperature, as we already know. They can be different temperatures, and many viruses can even be heat labile. So let's say we do need to kill them, then based on the property of the virus, let's say there's a temperature that they cannot survive in, then heating it to that particular temperature might be a way to kill the virus. But from a practical standpoint, you can see how that might not be possible. So there are also particles which are resistant to cooling. So these can either be inactivated or stored through, either dry freezing or lyophilization. So again, I've mentioned that they are obligate parasites because they are rendered inactive outside of a host cell. A few other properties which are not mentioned here is that, I'm sorry, actually go on to the next slide. Sam, next slide. Yeah. So sorry, I thought it was all in one slide. So like I've mentioned, the viruses have either RNA or DNA, right? So this RNA or DNA is what is responsible for actually encoding the proteins and which make the protein coat. And the protein coat is obviously very, very important because without it, the virus would not be able to exist, right? Without the protein coat, it's just genetic material. So basically, let's look at the case of like RNA viruses. So a slight point of difference between RNA and DNA viruses here. So RNA viruses, they actually multiply with a much higher speed as compared to DNA viruses. So this actually makes them quite dangerous. Why? Because faster the replication, less is the accuracy, which means that the RNA virus has a higher chance of developing mutations. And in that case, let's say even by some kind of infinitesimal point, if the mutation benefits the virus and greatly does not benefit the host, then it will be very dangerous for the host, as seen in, let's say, even the Delta variant and so on. So another point of difference is that RNA viruses have RNA-dependent RNA polymerase, so it's their own replication enzyme. Whereas for DNA viruses, they do have to use the cell's DNA polymerase in order to make new copies of themselves. So they are quite dependent on the cells. And since DNA polymerase has a proofreading ability, there's also a lesser chance of mutation, right? Now, we're also going to talk about, so another just small fact is that the mutations can also lead to the formation of different strains, as we have seen many times in the case of COVID as well. But if you look at just plant viruses, just the tobacco mosaic virus has over, I think, 50 strains. So that obviously makes it pretty dangerous. So before we move on to almost the end of my part of the presentation, it's an important point to look at is what exactly does the virus do inside the host cell? So what reactions does it bring about? And what do these reactions comment or indicate about the serological relationship between host cell and virus? So let's move on to the next slide. Yeah, so we will be talking about that. Now, again, like I said, I have already kind of covered most of this in the previous slide, where I'm talking about, the differences between RNA and DNA. One main point of difference is that, like I had mentioned, the RNA viruses can actually, since they have their own RNA-dependent polymerase as well, they also can have their own proteins because the proteins can be synthesized by themselves. So most of the time, the cells that they invade, their genetic material is DNA, but then the RNA virus has RNA as a genetic material. So they cannot depend entirely on the cell for, let's say, production of proteins and enzymes. So because of that, RNA viruses, they encode proteins which are required for their own replications and their own mutations, but they still use the cell's machinery to do it. So next slide. I've already finished this. So. Yeah. Now, let's talk a little bit about the serological relationship between the virus and the host. So what do I mean by serological relationship? What is this reaction that comes about? So this is something that we all actually know quite well, is that once any antigen, but in this case a virus, is encountered by the body, then it results in the generation of an immune response. So this is what constitutes that relationship. So viral proteins can also, so they are mainly a part of, let's say, the protein coat, but they can also act as antigens if that is what the viral genome commands them to do. So it depends a lot on the genome of the virus itself. Now, these viral proteins are the ones which are the antigens. There's always been a bit of confusion where people think the entire virus is the antigen. That's actually not the case, as I found out a few days ago. The actual antigen is the protein that they release. So how do these viral proteins, they are detected by an immune response, right? So how do they evade that immune response? Why is it so difficult to actually, properly curb a viral infection? So there are ways that the virus can evade this immune response by avoiding the detections by the T cell receptors. Now, obviously, as we know, the T cell receptors play a very important role in the immune response. And if anything with them is disrupted, then there's a huge advantage to the virus itself. So, how do they, evade? It's by modifying the ligands which would have originally bound to these receptors. So, the ligands on the proteins and on the virus, which would have originally bound to the TCR, what they do is they in a way modify that, so the lock and key mechanism between the TCR and the binding would be kind of inhibited or disturbed, so it won't create the kind of response that's actually efficient. Now they can also target antigen presenting process by MHC molecules, so this again will inhibit the T cell recognition. Now, they can, so all the immune system basically works through chemical messengers called cytokines and interferons. So viral proteins can even block them, which means that the message that, there is another invading factor within the body, it won't even reach the immune system. So because of that, the viruses will remain undetected, and they can in turn survive for much longer, and of course cause much more damage to the body. So this is the end of my part. Next, I'll pass it on, or I'm open for questions as well.
Yes, yes. So yes, very good presentations, Manaswini, very detailed one, and many good and new things in the presentation, right? So my question for you, Manaswini, is that why RNA is less stable than DNA? Can you tell
Uh,
me?
as far as I know, so
Just
one of the main reasons DNA is so stable is because it has a double helical structure, whereas most RNAs are single stranded, right? So the other, stabilizing factor is missing. So because of that, it can make it a very volatile genome molecule.
okay. That's fine. I mean, this is one reason, but if we talk about other reason, it could be somewhere like DNA has a deoxyribose enzyme, whereas RNA has a
Right.
ribose enzyme,
Enzyme.
right? So
Right.
that means the presence of this hydroxyl group make it more susceptible to the hydrolysis, right? So that is also another reason. Okay, the another question could be why? I mean, which you said that there are only few genes or a few genes in the viruses which are antigenic. It's not the whole virus. You are right. You are very right. So can you just tell me that which gene is usually and mostly antigenic part in the virus?
Okay, ma'am. So, from what I researched, I actually found that it wasn't just the gene, but it was also, let's say, a gene that codes for something called a glycoprotein spike, which I
Mm-hmm.
even in coronavirus, right? So
Yes.
it depends on the virus, of course, but if the gene codes for that particular spike, that's the antigenicity of the virus. So they
Yes.
help the host cell even establish infection. So that is what I think is the answer.
Yes, usually the code protein gene in all the viruses is antigenic. So what happened earlier times? See, I'll discuss about the plant viruses also. When we wanted to develop an anti-sera against the viruses, so we were taking the whole virus, right? So
Right.
a whole virus was taken, and then we used to go for. But these days now, we just take the code protein gene and we just express it in some vector, and then after that we just inject, purify that protein basically, and then inject in a rabbit to elicitate an antigenic response. So code protein gene is mostly the antigenic part. So if we just take that particular gene, it could be somewhere 1200 or maybe somewhere, or even just the conserved region in the code protein. Maybe there are some these days some softwares are there in the online, they're available where we can find the epitope regions in a particular gene. So just taking those epitopes or just taking those things, we can go for raising in particular antisera, right? Okay, good, Manas. Yes, next student.
Ma'am, am I audible?
Yes, you're audible. Who is this? Yes, Sam. Yes, Sam. Yes,
Yeah,
Sam. Please go ahead.
so DNA, the genome of plant viral genome. So DNA viruses are relatively rare in plants compared to their RNA counterparts, but there are double-stranded DNA viruses, plant DNA viruses that infect the algae, such as the Phycodinaviridae, and they infect the lower species of plants as well. Algae. So these viruses are huge double-stranded DNA viruses, and they have genome ranging from 160 to 560 kb, and they can code for around 600 proteins. So when we talk about proteins, then as Manas already explained, viral proteins. Uh, yeah, so well, viral genome comprises of coding regions that expresses proteins required for viral infection cycle, movement through the plant, and then also recognizing the host and movement between the host, and also they have some non-coding regions. Well, so there's the first one is structural proteins, and structural proteins are like the code proteins of small viruses, the matrix, the core, the cellular proteins found in the membranes, the lipoprotein membrane, the nucleoproteins, etc. And then there are enzymes like proteases and enzymes that are involved in nucleic acid synthesis. So virus code for one or more proteins that have an enzymatic function in the nucleic acid synthesis, either genomic nucleic acid or mRNA, or even both. And then some proteins are also used for movement and transmission, and like specific virus coded protein is identified by for its essential requirement for cell to cell movement between in a plant, in a host plant. Then there are non-enzymatic role in RNA synthesis. The 5' viral protein, genome-linked viral protein, found in some virus is thought to act as a primer in RNA synthesis. Then the last is host cell recognizing proteins, and so basically bacterial and animal viruses usually don't. I mean, they usually need to recognize. They have this host cell recognizing proteins, and they use it for infecting the cells, like one infected cell to infect further cells. But this is not the case in plants because once a plant virus affects a host cell, say in a leaf, so it can move from cell to cell through plasmodesmata, which is cellular pores, cytoplasmic pores, and then vascular, and also through vascular tissue throughout the plant. But it's used when suppose a plant uses a vector, insect vector, then the plant would need the virus would need to cross various barriers like the gut or salivary gland accessory cell walls. So the next is control of plant viruses. So virus cause obviously a lot of losses, and it's very hard to control or treat the plant once it's been infected. So there are various ways, and the first one is removal of sources of infection in or near the crop. So it's an obvious technique that if a plant is infected, then it's obvious to remove it from that so that it prevents other crops surrounding it to not be infected. Then the second is living host for the virus. So other than the plants which are growing nearby, there could be other hosts like weeds, then like the perennial weeds or annual weeds that are grown in or near these crops. Then there are ornamental plants that are grown, some unrelated crops, even plants of the same species from previous crop cycle. So all these could be a living host for the virus and could be transmitted into the new crop that's going to be grown. Then the next is plant remains. So plant remains in the soil, or sometimes in greenhouses, the plant remains can be attached to structures which can mechanically transmit a virus and can act as a source of infection to the crop. Then the next is hygiene, and it's also an obvious control. And so I'll talk about tobacco mosaic virus or tomato mosaic virus. So these are mostly transmitted to human activities done during cultivation, and hygiene is obviously used in like they wash their hands and all. But like a test done by broad, like sometimes the viruses could also be attached to the clothes and then they might get transmitted. So like a test done by Broadbent and Fletcher showed that tobacco mosaic virus persisted for around 3 years in dark regions in an enclosed space. But when it got inactivated in a few weeks in daylight, so earlier they used to use chemicals and also dry cleaning and detergents with hot washing and detergents with hot water, but they also now used to like they dry the clothes in the hot sun. Then the next is virus free seed. So sometimes like if a plant is infected in an early stage, then like the infection could be of virus in the seed. So we basically use ELISA for this technique, and everybody here knows what ELISA is. So yeah, ELISA is also used to test for presence of virus in batches of seed. And yeah, so next is heat therapy. As Manu said, some viruses, like most viruses, are heat labile. So heat treatment is a useful method for freeing plant viruses, and this is usually like the temperature and time of treatment is might vary depending on the virus, but we usually use uh, hot water treatment instead of dry heat, because dry heat doesn't give an even heating for short treatments. So we usually use wet heat for this method. Then the next is chemotherapy. So attempts to treat infected plant material of virus by applying certain antiviral chemicals. Even though this method hasn't been much used practically, but it's still been developing. Uh, Chemotherapy, along with heat treatment, has shown some advantages in a few instances. Then the last one is also pretty obvious: plant resistant to vectors. So if a plant is made resistant to the vectors, then obviously the virus which is in the vector can't be transmitted. So next is uses of plant viruses. So obviously from the start we have been just discussing about how viruses are a threat to us and also the plants. So but it also has some uses like virus as gene vectors. So from the 1980s, virus like there was an interest in using plant viruses as vectors to introduce foreign genes into plants, and so we use polymervirus, and because it's obviously a double-stranded DNA genome, so like there are various advantages to using this. Because first, there is high plant regenerate. Sorry, there's a like this virus could replicate to a high copy number, and then there is no effect on plant chromosomal DNA, and the infectious nucleic acid in the virus could be applied directly to the leaves, so there would be no need to use protoplast, and yeah, and the virus could move throughout the plant, thus offering the potential to introduce a gene into an existential crop. Next is virus for production of small peptides. So production of small peptides is required for various reasons, and including acting as epitopes for vaccines and biologically active peptides. So there are advantages to do it with plant viruses too, because virus can produce a large amount of uh, the proteins in large amounts, and the less developed countries where there is a need for animal virus vaccines, where there would be limited resources, this could be used. Then there is vaccines may be also be given orally as part of like normal food supply. Then the virus will not infect the human or animal, because obviously it's a plant virus. Then the system is not subject to contamination by other virulent animal pathogens. Then the next one is interesting: virus in cancer prevention and control. So, adenoviruses are used as vectors, and they can be engineered both to enhance specificity and minimize unwanted effects while treating this cancer. It's still being like researched about this, but it's could be like in a few years. Then virus-directed enzyme product therapy, VDEPT, and this is therapy that targets cells that are inserted with an enzyme that can activate they can activate an precursor or an inactive form of a cytotoxic drug. So this active cytotoxic form of the drug is only produced when the relevant enzyme is present. So an example of this would be adenovirus expressing the thymidine kinase enzyme of herpes simplex virus, and it can be combined with the systemic administration of ganciclovir, which converts the thymidine kinase to its active form. So then the last is nanocarriers. This is also a technique that's been researched right now, but it shows positive results. So virions are used to transport cargo compounds, and basically nanocarriers with the viral infection machinery, they are able to like uh, transport this virus divide peptides or proteins, and these viral components enhance the binding and uptake into target cells. And so virus or virus-like particles, they are composed of recombinant viral proteins. So I guess that's the end of my slide. Yeah, and next would be Adish. So I'm open for questions now.
Yeah, okay, Sam. So the best part which I like was the uses of viruses, because when we talk about viruses, the first thing that comes to our mind is like they are causing harm, they are being like as a very bad pathogens. But there is a positive aspect to them, which you mentioned in that, right? That was really good. Can you name few viruses which are used as vectors, plant viruses?
Ma'am, I said viruses which are used as vectors because
Mm-hmm.
they and other than that, I'm not sure about, but
Plant viruses is a cauliflower mosaic virus,
yeah,
mediated vector, basically.
polymoviruses.
Yes. Yeah. And can you name any plant virus which has been used as a nanoparticle in a drug delivery system?
I don't
No.
think.
Cowpea mosaic virus. It has been widely used as in a drug delivery system. They have modified it into a nanoparticle, and then they are being used in a drug delivery system in a gene therapy, and those modern techniques. It is being widely used. Yes, good, Sam. A good presentation and a very good content. Yes, next student, next last student, I guess, for this one.
Yes, ma'am.
Yeah, Adish. Is this Adish?
Yes, ma'am. Yes, ma'am. Next. Okay. Now, so coming to animal viruses, it's a small infectious agent that is unable to replicate outside its living host. So basically, it lives to reproduce, makes it copies, and infect other host cells. That's it. And unlike other intracellular obligatory parasites, for example, chlamydia and rickettsia bacteria, animal viruses only contain only one kind of nucleic acid, which is either DNA or RNA, but not both. Uh, Sam, next. During the after infection, what the virus does is they basically utilize the host cells' metabolic energy, their enzymes, their organelles, which they themselves are unable to produce. And what they do is basically hijack the cells, host cells, replication machinery, and they manipulate it to direct resources towards building essential light blocks of new virus. So so basically the host cell acts as a factory for new virus, and this process will go on eventually until either the cell dies first or the resources gets depleted, whichever comes first. Uh, the extracellular virus particle is called a virion, whereas the name virus is reserved for various phases of the intracellular development. Next. Okay. Viral genome, irrespective of the nucleic acid type, is almost always either single-stranded or double-stranded. They're circular, and in the case of polyomaviruses, and linear in the case of adenoviruses. Single-stranded genomes consist of an unpaired nucleic acid, analogous to like one half of a ladder split down in the middle, and double-stranded consist of complementary paired nucleic acids, just like a ladder. And all the segments are not required to be in the same virion for the virus to be infectious, as demonstrated by the Brome mosaic virus and several other plant viruses. We can also classify viruses through the genomic diversity, like through the property of nucleic acid. There are many parameters. It can be DNA based, RNA based, or both DNA and RNA in the case of retroviruses where reverse transcription is used. The shape can be linear, circular, segmented. The strandedness is single stranded, double stranded, and double stranded regions with single strandedness. And then the positive sense and negative sense, and then both ambisense. Next. Among RNA viruses and certain viruses, DNA viruses, the genome is often divided into separate parts, which are called segments. And these type of nucleic acid is irrelevant to the shape of the genome. For RNA viruses, each segment codes for one protein, and they usually found together in the capsid. The virus of some families, such as those of Heparnaviridae, contain a genome that is partially double-stranded and partially single-stranded. Next. No, in 1975, David Baltimore was awarded Nobel Prize for his works on virology, and he proposed a classification based on the genomic system present in the virions. So what he classified the viruses into 7 classes: the double-stranded DNA viruses, single-stranded DNA viruses, the double-stranded RNA viruses, plus positive sense RNA viruses, negative strands RNA viruses. Then we have the single-stranded RNA viruses with reverse transcriptase with a DNA intermediate. Then the same thing with RNA intermediate. Next. So this is the basically the representation classification of the Baltimore classification. We can, yes, okay. Okay. So the control measurements which is required to have these viruses at bay is to, since we are in middle of a pandemic, we should be aware of some control measurements. Like, it's basically common sense. Like, wherever the situation may be, just try to identify which part of it can be the most uh, infective, or which can induce the which can act as a carrier for many microbes. For example, if you own a farm, if you have farm animals to take care of, proper vaccination and keeping them clean from parasites and keeping them free from infectious microbes is a priority. Then the same thing is for same thing applies for almost everything. Then, for example, even though some living organism, be it animals or humans, they die because of microbial infection. So what we can do at this stage is to make sure to give it a proper disposal, either by cremating them or by burying them really deep. Really deep, by that I mean around 7 to 9 feet deep. Then regular disinfections of your animal houses, your houses, and wherever you stay. Next. Next. Okay. Coronaviruses are a group of related RNA viruses that cause disease in mammals and birds. In humans and birds, they cause respiratory tract infections, and which can range from mild to lethal. COVID-19 is a disease caused by SARS-CoV-2, and can trigger what doctors call a respiratory tract infections. Uh, SARS-CoV-2, which stands for severe acute respiratory syndrome, coronavirus 2. It can affect your upper respiratory tract, sinuses, your nose, your throat, and lower respiratory tract, which includes your windpipe and lungs. It spreads the same way as other coronaviruses do, mainly through person-to-person contact. Infection range from mild to deadly. It spreads through droplet and it's not airborne. It's also one of the 7 type of coronavirus which are there, and including those which caused the MERS and SARS back then, which was pretty much very deadly. And the other coronavirus can most of the they cause most of the common cold, and which affect people throughout the year, and which is it's just a common cold. So yep. That's it.
Okay, Adhish, you spoke about animal viruses. Yes, a lot of things. Uh, okay, my first question for you is that can you just name one animal virus which was used in a crude vaccination technique when the vaccine for smallpox was discovered?
Uh, Crude vaccination technique.
Variolation was used in that Edward Jenner type. Now, so what was the animal virus which was related to the smallpox and was used?
Oh, ma'am, it's the cowpox. They belong to the same
Yes, it
family.
is the cowpox. So it was used in the
Yes,
crude vaccination technique. Yes.
yes.
And my next question for you is that, see, the coronavirus is an influenza virus, right? It is causing viral. So why this virus is not being treated as journal influenza viruses are being treated, or they just go away within the 6 to 7 days and we all become okay? I mean, we don't even need antibiotics. What is so different about this virus?
Uh, Ma'am, coronavirus. First of all, they possess envelope, and they're also very quick to mutate. But then also many influenza viruses do. Uh, uh, I think it's their ability to produce, even though they have very high lethality, but they are highly transmissible. And I think the ability to mutate very quickly and produce many variants, which can sometime undergo even the vaccines, is what is making them much more deadly.
Yeah, yes. Coronavirus is also like an influenza virus, but the difference is that, yes, the high transmission rate of the coronavirus, and the second most important factor is that the infections which are caused by upper respiratory tract infections, which are caused by coronavirus in a small period of a time, it does not happen in the influenza viruses. You after some time you're fine. It does not go that. Quickly and easily to the respiratory tract, right? It doesn't reach your
Yes,
lungs so quickly. So this
and
virus
it's
do
just a
that.
common cold with
Yeah,
few
it's just a
symptoms.
common cold. Yeah, sometimes you may have congestion. That's fine, but you're fine after that. But in coronaviruses, it is multiplying so fast. It is going straight away to our respiratory tract, upper respiratory tract, and it's blocking there. Like pneumonia kind of situation is created where
Yes.
people are feeling suffocation. They are not able to breathe properly, right? So this is why it is. Okay, so we can take a short break, maybe 5 minutes, and then we'll come back, and then questions from other teams will a group will ask this group after the short break. Will after 5 minutes. It is 12:13. After 5 minutes, we'll join back, right? Thank you.
Thank you, ma'am.